where we are from yesterday. So we talked about an introduction to spontaneous generation. This idea that living things could come from non-living things. And people believed this for a long, long time. And they believed this because that was one way they could explain certain things that they saw. Like maggots just appearing on meat where there were none before. Or bread turning into this living mold organism. Or in the spring, frogs just emerging from the mud. So they explained all of these things that they saw by saying, well, given the right conditions, meat must turn into maggots. And bread turns into mold after a certain period of time. So we're going to talk today about some of the experiments and scientists that help study this and help us understand that there are other explanations for these things. One of the most famous was an Italian scientist, Francesco Redi, and he was doing his research in sort of the mid to late 1600s. And Redi was studying the problem of where do these maggots come from? Now, if you take a piece of chicken, raw chicken, and you leave it outside, a few days later, you come back and look at that chicken, it's going to be crawling with these little worm-like creatures, these maggots. And so Redi was trying to say, okay, well, where do they come from? He had his own idea. Most people thought it was the meat turning into them after a period of time. But Redi had a different idea. His hypothesis is that these maggots actually came from flies. So Redi did an experiment. He took 3 different jars, and in each jar he put a piece of raw meat. One jar he left just wide open. One jar he covered with sort of netting, gauze netting, so that air still could get in, but that was it. And then in the third he completely sealed it, so nothing could get in or out. And he made some observations. What he observed was that in the open jar, he noticed flies would be buzzing around, attracted to the meat. They would go inside the jar, they'd land on the meat. And what he saw is that they would lay eggs there. And eventually, maggots appeared on the meat. In the gauze-covered jar, again, flies were attracted, but they couldn't get into the meat. They could only get onto the gauze on the top. And so what he saw is that the flies landed on the gauze and maggots grew there on the gauze, but not on the meat itself. In the third sealed jar, okay, no maggots appeared anywhere. Okay, so what's his conclusion? Based on this experiment and these results, what is his conclusion? Ready?
Yeah, his conclusion was right because he said maggots come from flies.
Yeah, his hypothesis was correct. That these maggots were coming not from the meat, but from flies landing on the meat, laying their eggs there. And those eggs hatch into maggots. Maggots are a larva of flies. Just like a butterfly larva is a caterpillar, a fly larva is a maggot.
Who was the person who came up with this thing?
Oh, you mean the idea?
Yeah.
No real one person. It's just how people explain things that they couldn't explain otherwise. They just said, well, it makes sense, I guess. This meat, after a certain amount of time, just must turn into maggots because there's no maggots there before. And they suddenly seem to appear. So they didn't make the connection between the flies and the maggots.
Wouldn't he have gotten killed back then because of the people didn't want changes?
Some, I mean, you're talking about like Galileo, Copernicus, and things like that. I don't know if Redi was challenged because his ideas, they probably were not as controversial as some of the other scientists. All right, so that's one experiment. showed us it's not meat turning into flies, it's flies laying their eggs on them. Another scientist who studied this, so later, about 100 years later, was another Italian scientist, Lazzaro Spallanzani, and he was studying microbes or germs, or bacteria we would call them today, in broth. So what is broth? Michael?
It's like, I don't know what.
It is a liquid, yeah.
Oh, what?
How do you make like chicken broth? And
You make soup and it's flavored.
it's kind of like soup, but it's not with chunks of stuff in there. You basically take some water, put some chicken and stuff in there, boil it for a long time, take out all the parts, and then you have a liquid that has, dissolved parts of the chicken and stuff. And then you can make soup with it or just eat it. So what people saw was that if you had broth like that, you made some broth, you could eat it, it'd be great. But if you just left it, after a few days, it started to turn cloudy, started to smell bad. If you ate it, it would make you sick. There were these tiny little living organisms in there that caused people to get sick. And so they want to know, where are these coming from? After a certain amount of time, does the broth just turn into these things? And that's what they're studying. So Spallanzani, he's studying what causes these microbes to form in the broth. Question?
No, answer.
He had a hypothesis. His hypothesis was, again, basically against spontaneous generation. He thought it's not that the broth is turning into these microbes, but they're coming in the air that goes into this broth. So again, he did an experiment. In his experiment, he made some broth, and he put it in 2 flasks, kind of like these. So 2 flasks. What he did is he heated them both up. He boiled them. And then one of the flasks he sealed up, and the other he left open. So he boiled it, sealed one, left one open. The one that was heated and left open, after a few days, microbes started to form. Started to spoil. The one, though, that he sealed right away, it did not, no microbes formed inside of that. It stayed clear. A few days later, after he saw that it was clear, then he uncorked it, and a few days after that, microbes started to appear. So what do you think his conclusion was in this experiment? Karen? Yeah, that his hypothesis was correct. They do come from the air. Because as long as he kept the air out after he heated it up, the broth was fine.
Are we going to have to remember the names for the groups?
Yes.
Does it matter how we spell it? If we get it like close enough?
Yeah, that's right. So when we think about what we talked about yesterday, needs of life. All living things need a source of energy. They need water. They need gases from the air. They need proper temperature. They need a living space. So when Spallanzani did his experiment, he kind of took away 2 different things. When he did this initial step of heating up and boiling the broth, he was taking away the proper temperature because even those microbes, they can't survive that boiling temperature. So he killed them that way. What did he take away once he sealed the flask? Grace? The air coming in. All right. Our last scientist, he's not Italian. What do you think the nationality was? Louis Pasteur. Yes, he was French. He was, again, almost 100 years later. Louis Pasteur. He was studying, again, these microbes, these bacteria. And he used broth, again, as his media. And so he was saying, okay, well, where do these microbes come from? So again, Spallanzani showed they're in the air, but Pasteur took it a step further. He said, well, they're not just floating around in the air on their own. They're actually kind of hitching a ride on the little particles of dust that float through the air, that's where these bacteria really are. They're on the dust, not just floating through the air on their own. You ever been like at your house and sunlight's coming in the window and you see all the little specks of dust floating through the air? So Pasteur said, well, on each of those little specks there could be some of these very, very tiny bacteria. And that's where they're coming from. So he was pretty smart to this experiment where it was similar to Spallanzani. He took some flasks, but he made special flasks just for this experiment. These were called swan's neck flasks, where at the top of the flask, instead of being open, it came to a point, and then in a very narrow glass tube that made this sort of S-shaped curve. And it was open at the end. So it was this S-shaped curve, it was open at the end. And what that did is it allowed air to still be able to get into the broth. But dust that was in the air, when it started to move through, it would settle in the curve. All the dust would settle out. So in the curved area, the dust would collect, and then air could still get in them. So what he did is he took his broth, put it in these flasks. He heated it up, basically boiled it. And then on this flask, he just let it set. And air could still get in here, but dust could not. And this one stayed clear. It didn't spoil. No microbes formed there. On the other one, as long as he kept the neck of the flask attached, no microbes formed. But then when he broke it off, a few days later they did form. So his final conclusion was, he was right, that they are hitching a ride on these dust particles. It's not that they're floating freely through the air, they're on the dust. And so we know from these various experiments a few things, that this idea of spontaneous generation is a false theory. It's not correct. Living things can only come from where? Other living things. Life comes from other life. Now, for a long time, obviously, people didn't know these things. This is in the middle of the 1800s. So, but humans had found certain things that they could do that sort of would help them prevent these bacteria and stuff from becoming a problem. You know, in early Europe and other places, water came from streams, springs, things like that, wells. And sometimes those wells and those springs and those streams, the water was not great. It would often have in it microbes that would make people sick. But people noticed that if they used that same water that wasn't very healthy to drink, if they used it to brew beer, it didn't make people sick. There's water that they got did, but when they used it and made beer, it did not. So what was going on here? They actually would brew special types of beer called small beers, and they had low alcohol levels that they would give to children. Again, because this was healthier for them than drinking water that could possibly give them bacterial infections. So what was really going on here? Madison?
Well, they were brewing water to get rid of the bacteria.
How?
Was it like it was probably hot and it cracked?
Yeah, you're exactly right. To brew beer, you boil water along with hops and malted barley and other ingredients. And that process is removing what from the bacteria? What need of life?
microbes?
Well, why do the microbes die? They don't have the proper temperature anymore. So when you brew beer and you boil that water, well, all the bacteria are killed. And therefore, it's safer to drink that than to drink that polluted water, that water with bacteria in it. Now, you didn't really need to go through all the steps of actually brewing beer. What could they also have just done? Daniel? Just boil the water. And you may even know, like, if in your neighborhood there's like a water main break or something, you may get a warning that from the water company that says you have to boil water before you drink it. And it's called the boil water advisory. And it's got the same principle. There's a water main break. It's possible bacteria got into the water supply, and so you're supposed to boil the water before you consume it. Now, Louis Pasteur, you may have even seen his name this morning when you're eating your breakfast, or at lunch when you were in the cafeteria. Where might you have seen his name incorporated into another word?
And milk pasteurized.
Yeah, milk is what we call pasteurized. So what does that mean? It says on every milk, when you go, if you look at a gallon of milk in a refrigerator, it's going to say pasteurized. Comes from Louis Pasteur's name. What do they do, Rachel?
They boil the milk.
Yeah, so they collect milk, obviously, from cows. They bring it to a central area. And then they heat that milk up. They heat it up high enough to kill any bacteria. And then what do they have to do immediately afterwards? And close it, seal it up. And as long as they seal it up, bacteria aren't going to be able to get in there. And therefore, it's going to stay fresh longer. Canned goods, a jar of salsa or something, can sit on your shelf for years and not spoil. Because the process when they bottle that, they heat it up, they seal it so nothing can get in there. And so there's no way for bacteria to grow inside of that. But once you open it up, what does the package say? Refrigerate after opening usually. Because once you open it up, what can get in now? Bacteria and dust particles, and then they can start to grow. Putting it in the refrigerator slows down that process, so it lasts longer. But you know, once you open it, it's not going to last for that long. So these ideas were incorporated into how we make sure our foods are safe.
Does no honey like stay fresh for like 30 years or something?
I don't know,
Because
does
it's,
it?
like,
If it's sealed?
Capsulized or something?
Could be, I'm not so, I don't know about that.
I'm watching the video.
Yeah. Any other questions? All right, what we're going to do is we're going to watch this, just a short clip about spontaneous generation. Then you're going to answer some of the questions from the next couple pages.